looking at maybe easier topic, is the application of markers. I will show some results or some research that we have done to unravel the origin of cultivated and wild potato species, using COS markers and sequence analysis with GbS. And the outline of those are the topics that I'm going to talk today. A brief introduction about section Petora and how we use the sequence, the DNA sequences or the sequence of genes to test previous potato classifications, and also to review some circumscription of potato genomes. Also, we will test some, if potato has a single or multiple evolutionary histories. We will see also how COS markers or GbS can help us to define wild potato species, and to infer also genomic contributions of the potato polyploids and hybrid origins of diploid species. And so I think all of you know that potatoes belongs to the, is a member of the Solanaceae family, and it belongs to the subgenus Petoti. And we have three sections. Section Lycopersicum, where we can find the tomatoes, and we have also two other sections. Tuberosum, that is different. The main difference between both sections is that species in section Petota can form tubers, and here is Tuberosum not. And those sections are the closest to the section Petota. And okay, why we study potato? First of all, we are in Peru, and you know the potato is originating in Peru and Bolivia. At least the hypothesis is between Peru and Bolivia around the Titicaca Lake. And besides that, we know that it's the fourth crop in production worldwide. It's the third crop used as a staple food, so that is very important, not only in Peru, it's around the world. And but potato, section Petota, is not an easy group. Why? First of all, we have ploidy levels between diploid to hexaploid. In wild potatoes, we have between diploids and pentaploids, and hexaploids, and in cultivated from diploid to pentaploids. But in all sections, look at the range, from diploid, triploid, tetraploid, pentaploid, hexaploids. Also, we have auto and allo polyploids. We can find high morphological diversity, but also there are a lot of similarity between some species. So the classification of species in section Petota still remain very difficult. And previously, researchers were working with morphological data, only with morphological data, Einstein signs, RFLP, for example, on some markers, cytogenetic markers. And COS in 1990, emphasized about the origins of potatoes. He postulates that the primitive potato, wild potato, had a primitive stellata corolla and with genome B. Then some species from with this genome moved to the South America, allowing the evolution of the genome A. First of all, we have the primitive rotata, advancing rotata, and then some of these species with genome A move back to Central America, allowing the allopolyploidization, and some polyploid species with different genomes appears. So he based all of these hypotheses based on morphological data. So the researchers wanted to test if this has a support, not only for morphological data, but also with DNA sequences. Next steps were in the 1990s until 97. You can see here from 1984 to 1995, people were using plastic DNA restriction sites. And that is a summary, a tree that summarized previous results or initial results. Section Petota was divided in 4 clades. In blue, that is important that you keep in mind the following. In blue are potatoes, wild potatoes from North America, Central America, Mexico. Okay? So in clade 1 and clade 2 were all the diploid species from those places with genome B. The only one diploid species from Mexico was Solanum verrucosum, but with genome A. All of these species here have genome A, all. Okay, again, Mexican, US, Mexican, Central American diploids, and in clade 3, a monophyletic clade, we can find all the accessions from series Piurana and some accessions from other series of COS series. And here the remaining South American species, all the polyploid species from US, Mexican, and Central American in Solanum verrucosum. But as you know, this tree is the maternal tree. We are only seeing one piece of the history, the evolutionary history. This is a maternal tree. In order to use this tree as phylogenetic tree, we needed to use nuclear genes to verify which are the contributors of the polyploid species. The maternal side, we can see here the maternal side, we need to know also the paternal side. In that moment, we are talking about the 1997, people are starting to use different single copy genes to also to get insights into the evolution of potatoes. But they were finding different stories, different trees, no? With almost each single gene. But so we were thinking, what is going on here? What are the possible reasons that we can get different trees? So it can be technical issues, okay? That we can work on that. We can avoid those, but those we cannot avoid. Organismal level processes like rapid diversification, linear sorting, or resultant gene transfer, for example, and other gene and genomic levels, no? Intragenic recombination, all of this. That is in theory for other species too. It works for other species too, or it explains results from other species. But what happened in section Petota? Section Petota, we have a lot of integration. We have interspecific hybridization. As we say, we have auto and allo polyploids, sexual compatible among many species. Mixture, in the field you can have mixture of sexual and asexual reproduction. All of these problems. Then we started to use several single copy genes to determine or to get insight about the potato evolution. That's a result from two single copy genes, the boxy gene and the nitroreductase gene. And we were testing here previous potato classification. For example, COS says that North and Central American B genomes species, no, were basal, or were the basic, the more primitive species. So we can see here that, and also are monophyletic group, all of this. We also test if series tuberosum of Silicon shibacata were monophyletic, and it was, as you can see, the red ones, they go to different clades. Here is one clade and this other clade. They were not supported. And also, but it was supported the monophyly of Pimpinifolium and Polyadenium, no? All exceptions from Pimpinifolium and Polyadenium. All of those are diploid species. The other thing that we have tested here was if we can find some association between endosperm balance number if a species group together following the EBM. As you can see, there are some exceptions, okay? So that theory is not support to, no? And also it highlights some possible hybrid origins of two species, Brevicaule and Sparcipylon, no? Appear to have, because you can see that Brevicaule has some alleles here, and also Brevicaule and Sparcipylon. But in some way, work with diploids is very easy. Why? Because if this is a single copy gene, you can expect only two different alleles, no? It's a single copy gene and it's diploid. But in potatoes, in section potato, we have a lot of polyploids. So we wanted to know about what is going on with polyploid species. Next step. Now, those are the conclusions, but I already say that. So then we work with polyploid species, working with the same genes. And you can see that we test if the species in series Longipedicellata that are all polyploids that are really autopolyploids or allopolyploids. So Hertingi and Stoloniferum and Flenderin are species from series Longipedicellata. Some alleles are in this clade that has genome A. And other alleles from those species also cluster in this clade with genome B. So now you can see that they are allopolyploids. They have two genomes, A and B, and also we are confirming that Solanum verrucosum, that is a diploid from Mexico, remember, that has A genome, is the maternal donor of those species in this series. I will not tell you all the details, but it's the same in the other species, but you can see here series Piurana, for example, that is in pink, Solanum tuquerrensium paucicubum, and two species from this series, and alleles. You know that you can see here there are allele 1 from tuquerrense, here there are the other alleles, tuquerrense 1 and 2, paucicubum. All of them are in all clustered or grouped together in one clade. No? Those are autopolyploids. Same thing you can see here, those that are in light blue are species from series Tuberosum. All alleles are in only one clade. In green and polyploid species from series Conishibacata, you can see they have alleles here in clade 3, and also alleles here in clade 4 with genome A. So we use single copy genes to infer the genomic contributions of wild potato polyploids. But you see that those are few accessions from that species. We wanted to know if we test the same thing in other accessions of those species, we can get the same thing, the same result. Or maybe there are some kind of differences among them. Those are already the conclusions that I say, that those genes, nitroreductase and WOXY, are useful, but you will see that are useful, but not enough. No? Then we needed to get more genes. How we can find more genes that are single copy in those species? Because if they are not single copy, we will get a lot of alleles that are not getting the evolutionary history of the species. We will get a lot of noise. So in that time, in Cornell, we are talking about the 2006, our COS marker, that it is C for concern, O for orthologous, S is set of markers, were developed at Cornell from one student of Tansley. And basically what they did, they used ESP sequences from Arabidopsis, Tomato, and Potato, and they blast using the reciprocal base match for all of three. Later, they include also coffee and eggplants to get another set of markers that are supposed to be single copying in all of the 5 crops, no? But in that moment, it was only in Arabidopsis, potato, and tomato. They developed, or they identify, I think, around almost 2,500 COS. But those are supposed to be single copies, because you know that it's in silico. The identification is in silico. We needed, first of all, to test if they are really a single copy or not in potato. Second, when you look at the ESPs, you are looking at the expression sites, no? But for potatoes, we don't have a lot of polymorphisms in the exons of the genes. The region that is more useful for phylogenetics is the introns, okay? So we needed to find COS that are blanking a lone intron, okay? Then what we did, first of all, chromosome coverage, because sometimes we get the same results, the same phylogenetic, the same evolutionary history, because we are using genes that are in the same chromosome or in the same region. So those are not useful. First of all, we needed to know to cover all the majority of the chromosomes. Second, that have to be single copy, at least in potato and tomato. The sequence alignment length for each COS should be no more than 1,200, and the intron and exon content. No? We test more than 100 COS, and we identified 12 that were very useful. I'm going to show those results. In the other cases, we get a lot of sequences, different sequences. This means we were guessing that those are not single copy, no? And then they were tested again in the TASSEL GbS, and they confirmed that they are not single copy, at least in potato. And so we generate molecular data using sequences from those COS with PCR and direct cycle sequencing and cloning when necessary. Okay. Well, I say cloning when necessary because we direct sequencing. We amplify, we get the PCR, and then we sequence it with the forward and reverse. When we get the sequence that was messy, we can see 2 or more, so then we clone, okay? Yeah, it was all another nightmare, because how many colonies you have to clone in order to get the 2 alleles in a diploid species? How many clones you need to sequence when they are tetraploid or hexaploid to get, or to have the likelihood to get all alleles? No? 40, 60, 80, 100, okay? So, in the case of diploids, we sequence 8 clones, 8 to 10, okay, 8 to 10. In the case of tetraploids, we sequence 20. In the case of hexaploids, 40, okay? But you will see the problems with that, with the cloning, okay? And then we did the phylogenetic analysis using parsimony, livelihood, Bayesian, and also the concordance. I think everybody knows maximum parsimony, livelihood, and Bayesian. But the only thing that I want to stress here is the concordance analysis. The concordance analysis is a way to determine what is the proportion of the genome that is supporting each clade. That is also important. Not the sites, because parsimony, livelihood tell you the sites, how many sites are supported. That is the proportion of the genome. Then, let's say, here I am showing some of the characteristics of some of the COS that we use. Look, the total length was between 700 to 1100. The intron content, the best ones are with the 80s. 80% look for potatoes. It doesn't mean for other species, okay, or other sections. For potatoes, with less content of introns are not useful. And you can see here that they map in different chromosomes. And I think you also know that traditionally, when you have data from different sources, in this case from different genes, from these different markers, how you deal with the analysis. What do? You concatenate all the data and perform one single analysis, or you look at the trees. If you concatenate all the data and run a single analysis, phylogenetic analysis, you are using the total evidence approach, okay? Because you are going to get the most, you think, or the theory says that you will get the most probable of the evolutionary history, okay? But it doesn't mean it's the only one, or it doesn't mean that it's the 80% of the, you are getting the truth, you know, or, okay? You don't know the uncertainties. The consensus approach, what do? When you use this approach? You run a single analysis with each gene, okay? Here is the total evidence. You concatenate all the data and get one tree. Or maybe three, maybe, but it's only one phylogenetic analysis. Because when you run a phylogenetic analysis, you never, maybe with maximum likelihood, yes, you get one tree, yeah? But with parsimony or Bayesian, you don't get only one tree, you get a lot of trees, and you get a consensus tree, okay? But you are using only one database, okay? When you do consensus approach, you run different phylogenetic analysis, one with each gene. Then what do? You see the tree, you see the clades, and you, let's say you count. This clade is present in all the trees, so that's real, okay? But you do one by one, no? And the other thing is the prior agreement approach. You run a statistical test saying which of the genes are in which genes you are getting the same tree, or almost the same tree, and then you concatenate only those data, and then you run a phylogenetic tree, okay? No? This type of analysis was the traditional approach. In that moment, in Wisconsin, a new approach was arising. It was the concordance analysis. That is based in Bayesian analysis, no? And then you can get all of this with only one analysis, no? Using different values of alpha, with the hypothesis. For example, if we use alpha 0, you are saying that your hypothesis is that exists only one phylogenetic tree. If you use an alpha infinito, your hypothesis is that you can have one different phylogenetic tree with each gene, okay? So if you use different alphas, you can, in one analysis, you can get all the results, okay? Before, we needed to do separately. Then, here. As you can think, I couldn't use all the species or all the extensions that I wanted. It was a test. How many codes I have to use, or what are the best codes for 100, more than 150 codes we tested in the beginning. So, for the total evidence, we used 12 codes, and you can see the values here. Those are the bootstrap support when you use parsimony, likelihood, and Bayesian. In all of them, it's 100 or 99, because one is the probability. That is almost, in Bayesian analysis, you don't say 100, you say it's 1, no? That is almost like say 100, okay? More or less is the same concept. And prior agreement, when we did the analysis with the prior agreement, doing the test that I say, single analysis of these genes, we found that 6 codes, using 6 codes, we can get the same resolution of this. But when but if you see, we don't know about the other histories of potatoes. If you see here, you will say, oh, 100%, oh, very well resolved. That's the only one history. Okay? Then, when we ran, that is a summary. When we ran the concordance analysis, we saw that we got 3 different histories. That one is like the before, like with the total evidence, and we got the other 2. If you see the position of the species inside of each clade didn't change. The thing that is changing is the arrangement among the 3 main clades. Here, clade 1 plus 2 is sister of those 2. Here, clade 4 is the sister of the others, and here, clade 3 is the sister of the others. Okay. Those are the number of genes that are supporting each clade. No? Here is only supported by 4 or 3, 4 or 1, 3, 1 genes. Okay? So what do you think that is here, happening here? Is what is going on? Can be linear sorting, can be also that the number of genes are not enough, okay? And we were thinking, that is 12 COS are not enough for potatoes. We wanted to know if this history is going to change, if we include more genes, no? Then, because it says, concordance analysis is useful to summarize discordance among genes. Why? Because in one single analysis, you can get all the possible histories, and more genes could allow to discriminate between incomplete linear sorting and introgression or hybridization. Those sequences were also applied. Let me go back. Here there are only few species. If I remember, it was 12 or 11 species, I think. 4, 6, 11, okay. Then we applied the same COS, the 6 COS that were in concordance, we applied to define a species, to help to define a species. And now I'm going to show you only result from series Piurana, but we applied this COS for series Piurana, series Conoshibacata, y the Brevicaule complex. Those groups are very problematic groups because the morphology overlaps. It's not, sometimes there are not a single character that can define one species, or also the states of the characters. Some of the characters are present in all of them or in several of them. So we wanted to get insights also from molecular sequences to help us to define a species. We also apply for all more accessions of polyploid species and all wild species, and also to potato, cultivated potatoes. A species in series Piurana was defined by COS if to have globose or ovoid fruits, coriaceous leaves, and moniliform tubers. These three characters define series Piurana by COS. Okay? But when people ran the analysis using morphological data in all the species of series Piurana, they found that also some of those characters were in a species from Conoshibacata and Mexistracorolova. So the questions were, is series Piurana a natural group? What is the morphological definition and its taxonomic limits of this series? Relationships with other series, as I am saying, how they are related with the other series, like Conoshibacata, that are very similar, or maybe they are only one series. Which species should be included in this series? Are the species in this in Piurana monophyletic? This means Piurana is a monophyletic clade. Are they morphologically supported? All of these things were tested, and in the beginning, they have 40 taxa. Okay? After the analysis, 3 of them went to clade 4, and they belong to the other series, Konishibakata series. And 37 remain in clade 3. Clade 3 is the clade of the series Piurana, okay? But only, as you can count here, 20 plus 3 plus 6 are 29 species were recognized. And I'm going to tell you why. For example, some species based on their evolutionary history, of the monophyly, because those were formed monophyletic clades, and can be distinguished using taxonomic keys. For example, here, Solanum albornozi and Solanum olmosense. All of albornozi here, allele A, for example, here, allele B, allele A, allele B, all of these are monophyletic, and olmosense here is a distinct with a bootstrap of almost 80 or 85% with parsimony and livelihood. And they can be differentiated using this characteristic of the leaf. So, both species are confirmed being different. Next, Solanum albornozi and augusti. Here is Solanum albornozi, and here are Solanum augusti. Those grouped in different clades slightly differ in the shape of the fruit. Slightly. How you know if they are different species or not? Now we know because you can see very clear that those clustered in different clades, those grouped in different clades. So we recognize 2 different species, albornozi and augusti. Here, Gus masense and Ipacartrum. Okay? They are almost the same. We cannot differentiate, that is slightly different, and they also class together. And now there is only one species. We recognize only one species because also we are getting the support of the molecular data. And here, Solanum chacoense and mochiquense. Now they are lumped in like Solanum mochiquense. Okay? That's the use of sequences, DNA sequences, also to help us to infer taxonomic status. Then we applied the 6 COS in 44 polyploid accessions and in 37 diploid accessions to confirm the allopolyploid origins or autopolyploid origins of some of the species. But I told you that what happened when we were sequencing polyploid accessions? We needed to clone. I say that we cloned 8, we sequenced 8 colonies when we were diploid 40 or 60, remember? But we realized that we were getting also PCR recombinants, because when we did in that way, we were getting, for example, if it was a diploid that is heterozygous, we were getting maybe 3 alleles, different alleles, or 4 different alleles. In the case of tetra, it was the same. You will see, I'm going to show you some of the results. So we needed to move to SSCPs. It is the single strand conformation polymorphisms, okay, in order to separate the alleles without cloning. What we did the PCR as before. We ran the PCR in polyacrylamide gel, the natural gels, and we separate the alleles. But you see that we get a lot of bands. In the beginning, we take out the bands, all the bands, and sequence them. But it's time consuming to do all of this, and we wanted to reduce the number of bands that we are going to sequence, so we use asymmetric PCR. In this case, I am showing results running the same, running in polyacrylamide, the natural polyacrylamide gels, using the forward and the reverse. And you can see here is a part of this, no? see one of the, there are two alleles, one and one. That is the complement of this. So we only sequence one of them, no? Now here, for this accession, you sequence only this. Here you know that you have two alleles, one and two alleles. For this accession, that is one allotetraploid accession of Solanum kertingi, that is tetraploid. Look, we got two alleles from SSCP. And here I am showing only the polymorphic site. It's not all the sequence. It's only the polymorphic site from one COS. Okay, so this is one allele, and this is the other allele. So here is in bold. But when we did the cloning, in this case, we got only one sequence from cloning, clear and clean, okay? That it matched very well with the allele from SSCPs. But here, from cloning, we got two different alleles. One that match exactly like the allele got from SSCP, and the other is one that we think that is a PCR recombinant. Why? Because the beginning, look, is the same as the allele B, then is like the allele A. And things get worse. In the case of Eopetalum, that is an other tetraploid species, in one accession, we got from SSCPs three different alleles. Here are in bold, okay? And I can show you, for example, there are alleles that were cloned, obtained after cloning, with some tag errors that are small, like here, for example. But here, you have a PCR recombinant with sequences from the C allele, A and C. Look, here is the same as the C allele, that is here. Then, like the A allele until here, and then the rest is like the C allele, the last one. But when you perform the cloning, you will get 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So maybe 18 different sequences from allotetraploid. And you will think, oh, maybe this is not single copy. But the problem was not that it's not single copy. The problem was we were getting PCR recombinants. I am telling you that this is time consuming too. Less than do cloning, perform a cloning, but it's still time consuming. But until this moment, we really cannot sequence polyploids. Because if you know, I think you already talked about that, for example, sweet potatoes and potatoes that are polyploids are already sequenced. We have the genome sequence of those crops. Those are polyploids, but they were not sequenced, the polyploid accession. What were sequenced? Yeah. And a monoploid, in the case of potato, a monoploid, or a diploid that is highly homozygous. We are losing a lot of diversity, a lot of heterozygosity, okay? So until this moment, even now that we have different tools, with certainty we cannot avoid this process if we want to have all the alleles, okay? So we also confirm that, for example, Solanum, Eschenki, and Fugazi are composed by genome A, B, and P. Those are allopolyploids too. Eschenki and Fugazi are in red. Eschenki have alleles here in B. Eschenki in P. Eschenki in A. Those are allopolyploids with 3 different genomes. And Eopetalum also has genome A and P, no? Eopetalum is in blue. Blue, P, blue, A, okay? as you say, even that we have maybe 100 COS markers or 200 COS markers are not enough. We need to have more gene sequences or sequence the genome, but we cannot sequence 300, 400, or 1,000 accessions, okay? So we use GbS in order to reduce the complexity of the genome, no? And to sequence them. Okay, yes, you can sequence, but you don't have enough money to sequence them, okay? That's the truth, okay? So we are using now GbS. In the case of cultivated potatoes, previous studies were using here AFLPs, and as you can see here, there are not resolution. They are not well supported. Okay, the clays are not well supported, even with AFLPs. The only thing that you can say here is that data results are suggesting that probably the ancestors of the cultivated potatoes that are here are in species from Brevicaule complex that are from the north, not from the south, okay? Because here are the Solanum Brevicaule groups from the south, and here are from the north. That's the only thing that you can say, that results are suggesting. But with those bootstrap values, you cannot say more things. Then, Spooner in 2007 used 50 microsatellites, okay? Those are not sequence data, it's microsatellites, in almost 800 land races from the gene bank of SIP and 8 wild potatoes. And the same thing, maybe here, all the Solanum andigen and Solanum tuberosum, no, are clustered in here. But the bitter potatoes that are triploid, triplon, and pentaploid are all together, and those are 3 different species. Chaucha is here, but mixed with the Stenotonum, Pureja here, and some indigenous here, as you can see. We can say nothing. Even that the authors say that they can define 4 species here, but really we cannot keep 4 species here with this at all, but they say it, okay? Then, Cox in 1990 identified 7 cultivated species. Spooner in different years reduced, no, the 7 cultivated species, saying that he recognized only 4 cultivated species, and he wanted to test that. And in the beginning, we wanted to know which species, wild species, are the possible contributors of the, or what are the wild potatoes that from which wild potatoes the cultivated potatoes arise. Why? Why they are useful or useful for us? Why? Because probably it's easier to get genes from that species and to move to the cultivated potatoes and not to make speed the breeding. Okay, that is the main reason that we wanted to know that. Then we used GbS in 2011. I think all of you are familiar with this publication. Okay, then in order as I say, we cannot, for money issue, we cannot sequence all the accessions that we want, okay? One way to get more markers and to genotype in the same time is to reduce the complexity of the genome. We use enzymes in order to reduce all of these sites and to have a small site that we can sequence in the easy way, fast and easy. So, for example, we have sample 1, sample 2, you use one enzyme, that enzyme cut here, in all these vertical lines, okay? But in this sample, they don't have this place. The restricted enzyme is not recognized in here. Then you use a forward and reverse primer, and you will get a sequence of this, of this, of this, of this, of this. Here only this, here not sequence, here yes, yes, yes. It doesn't mean that the sequence inside is the same. I am saying only that the restrictions here are in the same place, they have the same sequence, okay? I'm not saying that all of these are the same here, okay? Because we want to find a SNP in that point, okay? Our hypothesis is that we will find a SNP here. There are differences between those. So, in the end, let's see. Let's say that I am talking about this piece of DNA, okay? That piece is this. Here we have the restrictions of APK1, here also that enzyme. It was the sequence, the DNA sequence that I separate or identify, okay? Then we have a DNA barcode for each sample. What is that? It's really a sequence. Sometimes it's from 4 to 8 base pairs that are unique and identify each of our accessions. And you will see why we need that. Then we have an adapter that it will be used as a forward primer and here as a reverse primer. In each well, let's say that in each well you have one sample, one DNA sample. 96 different DNAs, all of them. No? You digest the DNA with the restriction enzyme, you need to ligate the adapters, and you pull DNA, all the samples, you pull in only one. You mix all in one tube. How you will recognize it, and then you will sequence all of this. How you will recognize that one of the sequence belongs to this, the other sequence belongs to this accession. That sequence belongs to that accession. What? How? You will recognize with this DNA barcode. That's the thing that why we need the barcode. Then, okay. Those are steps that I never did because it's cheap and easy to send to US or to Europe, to China, actually very cheap, and they will do for you. You need to send the DNA, okay? And they evaluate, they look for the best enzyme for your crop, and you will get the sequence, okay? When you get the sequence, what you will do, you will get that sequence. You will need a software. The things that you are learning here, no? And the first software for this, it was the TASSEL. That also was developed in Cornell. That is a free, it's a free software, but it's not perfect. I will tell you later the things that we found and how we were solving that problem. But later, Reinhard is going to tell you more details about the software, but that is my understanding, so maybe it's going to help you, no? Remember here, I was telling you that this is the, let's say, we were talking about this piece of DNA, that let's say that this piece is here, okay? Then we have the enzymes, the barcode, and the forward and reverse. Okay? Remember all of this. That is one piece from one DNA, no? From one accession. Okay? Now, we will get this. All of different codes here, colors here are the barcodes. Each color is telling me that the sequence that is inside comes from this accession. Okay? Then, after we get the sequence, we will have a bunch, actually more than 500,000, like sometimes close to 1 million sequences. Then you will see that you have the barcode, you have the restriction site, the sequence, here is the sequence of the piece of DNA, okay? TASSEL only use 100, the first 100 base pairs of the sequence, and they discard the rest, okay? They didn't pay attention if this sequence here is reliable or no, if this sequence is okay or not. Because sometimes you get nothing, or trash, or sequences that are not useful when you get sequences. So, but they use, okay, now after you get from one line of sequence, this is another line of sequencing, okay, then here you merge all the tags here, and you count how many of them are different. How many, because many of them are equal. How many of them are different? and then you have a master tag count with the different tags, not different sequences, because you really don't know here the sequence, because it comes from when you align this with a genome of potato, for example, no? Then after you align, you will get the chromosome position, no, of that tag. Look at here, please see that here in this file, you don't know if this tag belongs to what accession. You don't know. You only know here how many or which one are all the different tag counts, no, that you found in all of the line of sequences that you got. After this, your master tag count that comes from the other slide, you will compare, and you will see to determine the distribution of these master tags among your samples. For example, this is the first tag counts that you found, and you will see if this tag is present on the first accession. Is present? No. Present? No. Present? Yes, only one time. Absent, absent, absent. Present 3 times. Present 2 times. Da, da, da. Like this. Okay? Next. About the filters that you can use, Reinhard is going to tell you about the filters. After this, you use that file plus that file that we were talking about, plus the file that has the tags with the position of in the chromosome, the position of your tags, and then you will get this, they call production ready file with all of the tags with the positions that you get in the in all of your accessions, no? Then you will use this file to call the SNP in your accession, okay? Look at this. In this moment, we are not saying that here we don't know yet if here we have a SNP or not. We don't know how many SNPs we have. The only thing that we know is that we have the same position in several accessions, let's say. But we don't know how many SNPs we have yet, okay? That is, Reinhard is going to show you how to do that. As I was telling you, really we didn't realize that TASSEL has problems. In my case, I'm not a bioinformatician. I do my best effort, but I didn't realize that TASSEL has problems. How we were thinking that TASSEL has some problems. Why? Because people that have already mapped and know the people at SIP, in that moment I was at SIP, people that know the almost the exact position of some gene, because they work with mapping populations, they already know the position of some genes. And they use TASSEL and they use GbS, and that marker was mapping in different positions, you know? And they were saying, what's the problem? Something happening with our population. Maybe in the field they were missed. What happened? It was a big problem, really, because we were thinking that the problem was in the field. But then they realized that the TASSEL has at least had at least three problems. First of all, blind read filtering. What does mean that? Remember that I say that the sequences of the pieces of the we were talking can be maybe 400 base pairs, but TASSEL only used the first 100 base pairs, okay? They used only the first 100. Yeah. So without paying attention is maybe they have reliable sequences until 200 base pairs, for example. What does mean that they are, first of all, they are losing information. They are probably losing useful information. That's the correct way to say it. Second, each sequence, okay, was not attached with some predicted value about the quality of the sequence. That is also very useful to know. In that moment, we didn't have that values. And the worst was this. They were ignoring mapping information. when they map, the software only recognizes the first site where the tag map. Sometimes the tag can map in different places, or at least in two, and they forget about that. They even try to find the other. After they found the first, the software stopped, you know? But thank God that it happened, because that's the only way that we realize that we have some problems with that software. So, Alexander contacted Reinhardt. He's a bioinformatician from Germany, and he came to SIP to try to help us to resolve this problem. And he developed a variant pipeline that was solving those issues, you know? And then we ran all of our analysis using the pipeline of Alexander. And he built that in Galaxy, and you know that Galaxy is very friendly. So it's, I will not say easy, but it's easier than other softwares, okay? And then, okay, the only thing that I wanted to show you here is why we were using GBS, because I told you that we wanted to know which species are the possible progenitors of the cultivated potato, no? Okay? So we use some wild species, okay? Some of those were hypothesized for other taxonomies that are the closest to the cultivated potatoes. And here, Stenotonium, Gonocales, Cureca, Nickel, and Tuberosum, all of them are cultivated species. From here to here are all wild species. And you see that Bucasobi, Candolianum, and Ambocinum, all of them cluster with the cultivated potato. But, so now we have more insight that if those species are the progenitors, and right now we want to use more accessions from those species, because here we have only few. We want to use all the accessions that are in the gene bank from these species to confirm if really those species are the closest. Or maybe Bucaso or the closest are accession from this species but grow near to Titicaca, because we saw some pattern. The species accessions that are close to the cultivated species are from Puno, then from Ayacucho, and then from Apurímac, okay? But in this moment we don't know. But the only thing that we wanted to show you is the utility, how we use these markers also to solve some scientific problems, not to test. And also, the other thing that I wanted to tell you is that even we don't understand very well bioinformatics, like me, we can use and we can have so if some of you feel it that maybe bad, that is too difficult, whatever, but little by little you can get it, no? The problem with the GBS still is the missing data. The amount of missing data is very high, okay? But we are using only variants with less than 5% of missing data. We run different analyses, using sorting or choosing SNPs in different ways, okay? But finally we were using only SNPs with, that have less than 5% of missing data. Yes? Yeah. Thank you for saying that, because I forgot something. Here, all are diploids, because this is a very good point. And Stenotonium is diploid, you know. The only tetrabords are those, okay? But basically, only because we wanted to see, good point, if that complexity for the tetrabord was okay. If you see there are few accessions, only to have some idea, but we were really focused to know about the evolution of the diploid species in relation with the wild, okay? The other thing that we can discuss too, but we don't have the answer, is about the plexity. In the case of diploid species, we know that 96 plex is good enough, but in the case of tetraploid, we use 48, but it will be enough or not? 24, 16. If we work with the other wild potato polyploid, remember that we have hexaploids too. What will be the plexity? 12? We need to test, I am saying. We don't know in this moment, okay? Then, there are many things that we have to do. Still, there are a lot of things that we have to do. Then, the other thing that we wanted to make sure is the chromosome coverage. If our SNPs are in all the chromosomes and the chromosomes, the regions of the chromosome well represented, okay? So you can see that this is from chromosome 5. But you can see the same picture almost in all in the 12 chromosomes of potato. Another thing that is important to us is the heterozygosity, especially for breeding. For many markets, you don't get high heterozygosity in diploid species. And when people were using the array from Illumina, because there is an array, there are 2 arrays from Illumina that are available in the market. But you know the arrays are easy. The analysis, the informative part is very easy, but those are very expensive in comparison with GDs, with other technologies, okay? So people that are using arrays are not getting that levels of heterozygosity. Why? Because the arrays are based on few genotypes, as you know. And at least 4 gene band accession, different species, those are not useful. Those are useful for other things, mapping population, for example, no? Yeah, but not for diversity. And the important thing that I think the people that work in potatoes will appreciate that is that you see with the SNPs from arrays, they were getting for this species heterozygosity of 1%. Here we are getting 6%. No? And those species that are in red are the Ambocinum, Bucasobi, and Candolianum that we found that are the closest of the cultivated potatoes that are here, and look at the heterozygosity, 10, 13, and 17. Close, very close to the cultivated potato, the diploid, no? Because the tetrapods are more. Okay? Future steps. What has already been done? Optimizations of the workflow to improve the variant calling algorithms. We are using also other reference genomes for the mapping, the commerce zoning. We also are identifying indels and use those in the phylogenetic analysis. But we still need to explore better software to estimate ploidy and dosage in the polyploids, and to get more representation of the other species, no? The triploids, pentaploids, and hexaploids, okay? And also, not only to work with accessions from the gene bank, also from in situ, because accessions from the gene banks were collected in the 80s or in the 60s, and in situ, the evolution is happening right now, and we want to compare with that too, okay? And then I wanted to thank our collaborators, and that's it. If you have questions, no, you can say in Spanish. No, okay.